We're going to talk about population and community ecology. First, let's talk about what we mean by population, and tangential to that, population density. So population density is the number of individuals of a particular species per unit of area or volume. So notice that we're focusing on a particular species. That is what we mean by population ecology. And population density is useful when comparing two populations of the same species in two different areas. Now, those areas we can consider to be anything that is focused on our question at hand. The boundaries between those areas could be multi-regional. We could be looking at the international spread of population density here as imaged on the far left graphic. Or we could be taking more of a snapshot of regional differences in population density, as is the case in population density in human populations, for example, in the United States here. Or our questions could be more local. We could be asking questions about population density on a more local scale, and a more local scale would be represented by a state, for example. Even within the state, we could focus in on a county, we could focus in on a city. So all of this is to say that population density questions really center around what are you defining as your boundaries. Once you have your boundaries established, next question is how are you going to sample that population? We in science use multiple different sampling techniques because it is really impossible to count every single member of a population. Some populations move around a whole lot. Some of them are so small you can't really see them. And just for the sake of time and for the sake of money, we take alternative approaches that are really robust for sampling different populations and their density. So a few of the approaches here. Number one, we have something called quadrats. And this is what is being imaged here in the upper right. This scientist is holding out a probably 1 meter by 1 meter quadrat. This marks off a square boundary at several locations. And in those several locations, you will take measurements of your species and then take an average. This is mostly useful when we're talking about population density of plants or population density of other sessile organisms that are affixed to the soil or sediment and don't move, like lichen. Next, we have indirect counting. In indirect counting, you are not counting individual organisms. You are counting evidence of the presence of those organisms. This could be things like bird nests or burrows or tracks. So all of that falls under indirect counting. Next, we have mark recapture. And this is when scientists trap and mark mobile individuals for future tracking. This is used for keeping up with populations of salamanders, of birds, of fish. And the way scientists use mark recapture is they use the ratio of marked and unmarked individuals during a second sampling campaign. And with that ratio, they determine how many individuals are in the sample at large. So from this, they're able to make estimates on the total population size. And this method assumes that the larger the population, the lower the percentage of tagged organisms that are going to be recaptured during that second sampling campaign. So I'm going to ask you, which technique would you use to estimate the population density of some of these organisms? So first up, we have this cute bird. Which do you think would be the most useful in capturing the population density of a bird? Well, on the previous slide, we discussed two different ones. We could use mark recapture, but a cheaper alternative is indirect counting, because that only requires one trip to the field. You can count all the nests one time, you're done. With mark recapture, you have to first capture, mark, let them go, and then come back at a later time and recapture them. What about something like a cactus plant? Well, the cactus plant doesn't move around, and so this would be best approached with a quadrat method. How about a bat population? We can already see that this little bat has a wing that's been tagged. And so this is a representation of an individual, part of a mark recapture experiment. Now, once we know the numbers, there is an additional layer behind that population density, and that's the population distribution. So just because you know the number of individuals, it doesn't say how those individuals are distributed within the habitat. So here we have a figure from your textbook representing the three main types of population distribution. First, we have a uniform distribution. This is most common around territorial birds like penguin species. Next, we have random. Random is where you can have the mix of a uniform and a clumped distribution. Some members of the population are clumped together while others are scattershot around. And this is most notably noticed in our dandelion species or other plants that are dispersed via wind. Sometimes a number of seeds get pushed to the same spot all at once, and therefore they colonize pretty much the same area. Other times, one individual seed gets dispersed far, far away from everything else. And next, we have a clumped distribution. This is exhibited by herding animals, mostly elephants depicted here, and it's just as it sounds. All of the individuals are clumped together in their respective herds. So again, I'm going to ask you, based on this information, which distribution pattern would you expect for these specific animals? So first, we have termites, represented here by these massive termite mounds. So what type of population distribution would you expect to be characterizing our termites? The answer should be clumped because they're all living in one termite mound together. Next, how about a rattlesnake? This would be a random population distribution. They would pretty much be scattershot everywhere depending upon resources and territory and food. How about a bird? This would be characterized by a uniform distribution. Next, let's talk about some of the changes that a population can undergo through space and time that affect some of these characteristics. First, let's introduce this term of demography. Demography is the statistical study of population changes over time. This is relating to things like birth rates, death rates, and life expectancies. And each of these measures, especially birth rates, may be affected by the population characteristics described in the previous slides. So for example, if we have a large population size that results in a higher birth rate because there are more potentially reproductive individuals that are present. In contrast, a large population size can also result in a higher death rate because of lots of competition, because of susceptibility to disease, and the accumulation of waste. Similarly, a higher population density or a clumped dispersion pattern results in more potential reproductive encounters between individuals, which could increase the birth rate. And lastly, a female biased sex ratio, meaning there are more females than there are males in the population, or if the age structure is such that it's mainly composed of individuals of reproductive age, this can increase the birth rate of a population. So all of this is to say that these demographic characteristics of a population can influence how the population grows or declines over time. And the mortality rate here we can represent in a population of any species to be the number of individuals dying divided by the number of individuals surviving multiplied by 1,000. So let's talk about survivorship and mortality. We have three different types of survivorship curves that are characterized by different types of animals. So type 1 reflect relatively low death rates early in life and through midlife, with a sharp increase in death rate among older age groups. So this type of survivorship curve is what characterizes human populations. We typically live longer, and the longer we live, the higher the chance of us perishing. So here we have on this chart here, the x-axis, percentage of maximum life expectancy, and the y-axis is the number of individuals surviving on a log scale. So here we have a human representing that type 1 survivorship curve. Here we do not experience rapid mortality early in life. When we get to about midlife, that's when things start to decline and crash as we get to that maximum life expectancy here. Type 2 illustrates a fairly even mortality rate throughout the entire lifespan of the individual. Here we have the example of birds, and we have this straight line. And this just goes to show that there's an equal chance of dying at any point in the organism's life. Type 3 means when we have populations with high death rates early in life, followed by a sharp decline of death rates for the survivors. This is what we see in fish, lots of insects, and many trees. So here again, here's our tree with type 3. There's a rapid loss of individuals early in their life expectancy, and then that mortality rate peters out. So these are the 3 different survivorship curves that can characterize different populations. Something that goes hand in hand with survivorship is something called life history of an organism. Life history describes a series of events over the organism's lifetime, such as how resources are allocated for growth, maintenance, and reproduction. So a species' life history is genetically determined, but it's also shaped by the environment in which it lives, as well as, of course, natural selection. We also need to introduce this term called fecundity. Fecundity is the potential reproductive capacity of an individual within a population. It describes how many offspring can ideally be produced if an individual has as many offspring as possible, repeating the reproductive cycle as soon as possible after the birth of their last child. So in animals, fecundity is inversely related to the amount of parental care that's given to individual offspring, meaning if the mother has to invest a whole lot of energy and time into raising one individual, it's not going to immediately seize upon the opportunity to have another child. And different populations have different levels of fecundity. For example, we have the example of our salmon, our antelope, and our chimpanzees. And all of them display different types of energy in terms of rearing their young. And this all comes down to something called an energy budget. Everyone must balance energy intake with the use of energy for metabolism, reproduction, parental care, and energy storage. So let's talk about how fecundity and the energy budget all intersect in an animal's life history. Specifically, the timing of reproduction in an organism's life history affects species survival. If you are an organism that reproduces at an early age, there's a greater chance of producing more offspring, but this is at the expense of their individual growth and maintenance of their health. So for example, small fish like guppies, they use their energy to reproduce rapidly. Okay, but because they're constantly reproducing, they never attain the size that would give them defense against some predators. And therefore, they remain small because they are dedicating their energy budget to increasing their fecundity and having more offspring. By contrast, an organism that reproduces later in life, they have greater fecundity or are better able to provide parental care, but they risk that they will not survive to that reproduction age. So they're waiting to attain a larger size, therefore allowing them to dedicate their energy budget for themselves, but they run the risk of never getting old enough to reproduce. And so large fish like the bluegill or shark, they use their energy to attain a large body size in their life. But of course, they do so at the risk that they will die before they can find a partner, mate, and reproduce and rear young. And so these different energy strategies and trade-offs are key to understanding the evolution of each species as it maximizes its fitness and fills its ecological niche. Next, let's talk about instances of reproduction events. You can be an organism that reproduces only once, or you can be an organism that reproduces multiple times throughout your life. Female parity occurs when a species reproduces only once during its lifetime, and then it dies. An example of female parity that you probably have heard about is the salmon. And salmon use most of their energy reserves to migrate from the ocean to its freshwater nesting area where it reproduces and then it dies. And this is a programmed suicide, it's thought, to provide offspring food as it needs nourishment for growth and development. By contrast to semal parity, we have heteroparity, and this describes species that reproduce repeatedly during their lifetimes. And within heteroparity, some animals are able to mate only once per year, but they survive multiple mating seasons. Some of them, like the pronghorn antelope, have to undergo this seasonal estrus cycle that we colloquially call heat. And this is a hormonally induced physiological condition that prepares the body for reproduction. And there are other organisms like humans and chimpanzees, for example, that can reproduce pretty much at any time. It's not a seasonal thing. We don't undergo this seasonal estrus. Instead, we have menstrual cycles that make pregnancy likely over a few days per month during ovulation. So all of this feeds into population growth. And a population's ability to grow partly depends on the rate at which its organisms can reproduce. The accelerating pattern of increasing population size is called exponential growth. So we can represent population size with n here. And a good example of exponential growth is displayed by bacteria growing in a culture tube. They have what is called a J-shaped curve. And this happens when there's no nutrient limiting their growth and reproduction. Instead, they grow at a constant factor at a constant time interval. For example, bacteria, depending upon the species, E. coli doubles every 20 minutes. So this results in a J-shaped curve. And we can define in mathematical terms the rate of population growth as the change in the number of individuals in the population over the change in time equals the biotic potential or the maximum growth rate multiplied by that population size. In contrast to that J-shaped curve, we can have an S-shaped curve. This represents logistic growth. This is more realistic because with logistic growth, there is a limit on nutrients and resources available. So we do start with exponential growth, but of course, one or more environmental factors are going to impact the ability of that population to continue to grow. And this all falls back on our economist, Thomas Malthus, with his idea of limiting factors in the environment, reducing the amount of individuals that can survive in a population. So this means that there is an upper limit called the carrying capacity for every environment, for every population. Okay, we can represent the carrying capacity with this term K. And K is the number of individuals that the environment can maintain or carry. We can represent in mathematical terms the logistical growth curve in this way with the introduction of a new term of K, the carrying capacity. So all of the terms remain the same from the previous slide, but this K, carrying capacity, is a very important one. It works hand in hand to define what population growth is going to be slowed by. So when N, or the population size is large, this term K minus N over K comes close to zero, which means that the population growth is going to be greatly slowed or even stopped altogether. So population growth is greatly slowed in large populations by this K term. We have different factors that are regulating population growth, and these are sometimes related to the number of individuals in the population and sometimes not. First, we'll talk about the ones that are. These are called density dependent factors. These are ones that are really contingent upon how many individuals are within a population. A good example of this is the fecundity as a function of a population within the giant intestinal roundworm. As you increase the number of worms within the environment, the number of eggs per female that can be produced falls off dramatically, okay? So this is showing that there's a specific relationship between birth rates and the number of individuals in the population. You can also have a relationship between death rates and the number of individuals in a population. And those death rates are typically associated with competition, both inter and intra-specific competition, predation. When there's more of you in a population, you are more susceptible to being preyed upon. Also, you are more susceptible to disease. Once a disease takes hold of a population with lots of individuals, it can rapidly spread. And you can also have lots of waste build up if there are too many individuals within your population. All of this works hand in hand to increase mortality rates at high population levels. By contrast to density dependent factors, we also have density independent factors. These are unrelated to the number of individuals in a population. We consider these to be extrinsic factors, things like temperature, humidity, photoperiod, how much sunlight do you get during the day. And so it's the combination typically of density independent factors and density dependent factors that ultimately result in a population's demise. And of course, nothing in nature is simple. And a good example of this is the interaction between density independent factors and density dependent factors with the woolly mammoth. So what caused the woolly mammoth to go extinct was the combination of climate change, a density independent factor during the Cenozoic that reduced their population numbers, and then when you introduce predation upon them, a density dependent factor from early humans that then drove them to extinction. So let's talk about how all of this rolls into life strategies. A life strategy simply is the regulation of population growth within a population that is balancing parental care and offspring output. So there are two main types, K-selected versus R-selected species. And these are again related to species reproductive strategies within their habitat. And it's a balance between resource acquisition behaviors, rearing behaviors, and length of life. Within K-selected species, we are within a stable, predictive environment. No rapid climate change, no habitat destruction or disruption. And so these species exist close to their carrying capacity, hence the term K-selected. They do have high intra-specific competition, so within the species, and they have few large offspring. They have a very long gestation period, meaning the mother is carrying the young inside of her for a long while. This is coupled with long-term care for the young because the young are immature at birth and they need a lot of care and protection and rearing. A good example of this is with the great apes. The mother spends a long time rearing that one individual, putting a whole lot of time, energy, and investment in raising one healthy organism that will hopefully attain reproductive age later on. In contrast to the K-selected species, we have something called R-selected species. These species live in unstable, unpredictable environments. They have a large number of small offspring, hence the R designation. No long-term parental care is provided, maybe a shorter gestation period, and these organisms are pretty self-sufficient at birth. Therefore, they don't need to be taken care of for a long while after the mom has given birth to them. So this all comes down to balancing output of offspring versus parental care. And so here we have an example of two K-selected species which live long, mature lives, and they produce few offspring. Here we have the R-selected species like dandelions or jellyfish because they have short lifespans and they produce many offspring that have no or little parental care. Again, K selected meaning carrying capacity selected, R meaning they're trying to boost their population growth rate. And here are some characteristics of the K and R selected species. You can refer to this on your own time. What we see here is a boom and bust between the R and the K selected species of different types. So with the K selected species outlined in blue, we don't see a rapid boom and bust, but we do have this very delicate, subtle ebb and flow of the population size hovering around this carrying capacity. By contrast, for our selected species outlined in red, we see massive boom and bust cycles. So boom when that population increases above carrying capacity, and bust when resources are depleted and that population declines. And so we have rapid boom and bust cycles within the R selected species.